Assalamu alaikum viewers, welcome to Academia Coaches. The topic of my today lecture is regulation of gene expression in eukaryotes. The eukaryotic gene expression is more complex than that of the prokaryotes due to the presence of nuclear membrane, and this nuclear membrane separates the translation process from the transcription. The transcription takes place in nucleus of the cell, while the translation takes place in the cytoplasm. The eukaryotic cell regulates gene expression at many different levels. The level of eukaryotic gene expression includes, number one is the accessibility of DNA. Number two, transcription of DNA. Third is translation. It also includes the post-transcriptional modifications and post-translational modifications. Today in my lecture, I will discuss about the accessibility of DNA. Then in my next lecture, I will explain about the process of transcription and translation further on. Okay, let's start with my tutor topic, that is the access or accessibility of DNA or accessibility of chromatin. The DNA in the nucleus is wounded, folded and compact in histone proteins so that it fits in the nucleus. These are the histone proteins. And the DNA is wrapped around the histone proteins in the nucleus. And these histone proteins package their DNA into structural unit which is known as nucleosome. The histone proteins package their DNA into structural units which are known as nucleosome. The nucleosome control the access of protein to DNA region. Nucleosome control access of protein to DNA region. If a gene remains turned off, in this case, the histone proteins, these are histone proteins, and the DNA. This is the structure of DNA. These are the tails of histone protein. Okay, when the gene turned off, then the histone protein and DNA have different modification, which is known as methylation. Then the methylation of DNA or histone proteins result in compact or tightly packed form of nucleosome. And transcription factor do not bind to DNA and genes are not expressed. And if a gene going to be transcribed or for the transcription of the gene, the histone proteins and the DNA are modified, which results in loose packing of nucleosomes. It results in loose packing of nucleosomes. And due to this loose packing of nucleosomes, It can be by the acetylation of histone proteins. And due to acetylation of histone protein, it results in loose packing of nucleosome and transcription factor bind to DNA and gene expression is regulated. Okay, I am going to repeat the case. When a gene is undergoing to transcription or transcribe, the modification in the histone, that is the acetylation of histone proteins, result in losing of nucleosome. It results in losement or losing of nucleosome and DNA packing. Due to losement of nucleosome and DNA, the DNA is accessible to the transcription factor and it results in expression of genes. Okay, this lecture was all about the opening or accessibility of DNA to the transcription factor, which is the first step in the gene regulation. In my next lecture, I will explain about the transcription of DNA, that is the copy of DNA into the RNA. Thank you so much viewers and Allah Hafiz. Do not forget to subscribe my channel for latest and more relevant videos. Thank you.